Today, we will be talking about bacteria. We'll be doing bread mold and also bacteria swabbing to actually help bacteria grow on a petri dish. And so, in your bag of materials, you should have at least a petri dish and some cotton swabs, maybe some hand sanitizer, because in between, we're going to be touching different things. And then you also should be able to have some bread and you should have 5 pieces of bread. I have bagel bread for my thing, but it's okay if you don't, or if you have regular sandwich bread, that's fine too. And you'll need some tape and some magic markers or a pen, and you'll also need the sandwich bag for it to go into. And today we'll be talking about fungi, which will be grown on your bread mold, particularly. So fungi are neither an animal nor a plant. Fungi do help decompose things, and they basically naturally enrich the soil with nitrogens and phosphorus groups because they're breaking apart things that have these minerals inside. Fungi are multicellular eukaryotic cells. What's fascinating about them is that you eat them maybe once in a while, unless you're allergic to them, which is mushrooms. And so you can see here, there are mushrooms, different types that you see here, and then some that are growing on our bread. I also want to point out that not only are they eukaryotic cells, we also see that yeast are as well. So when you're eating bread, such as if you're eating, a little small bite of a bagel like this, you're actually eating some fungi because the yeast that helps rise to lift up the bread is actually, a fungi and it's part of that family. Also, what's very interesting about yeast, as well as other types of fungi, such as mushrooms, they're super nutritious. Yeast have B vitamins inside. Mushrooms contain a lot of trace elements that we need to actually power our neurons. So, for example, manganese or magnesium, and then iron to actually help regulate our blood levels. And so all of these things are very important. And so it's very thoughtful considerations when we think about fungi to have respect for them because they are very important. The most fun fact is that they are decomposers. They break down organic matter. So today, when we actually are thinking about actually making bread mold, we have to think about how these pieces of bread could also decompose because of the mold that's on them or the bacteria, because we will pick up a couple of pieces of bacteria that are on the mold along the way when we're swiping them on the computer screen or on the side of our face or something. So I just want you all to be aware. So what are some common things we use fungi for? Well, we use them for mushrooms just to eat. We also use them when we're actually making clothing. And also we use them to bake different types of bread. And most of all, the ethanol that you use in your car, guess what? Yeast helps to make that. Fungi can be used for so many different things, and there's different classes of them. Now, also, what are some other common things that we use fungi for? Well, everyone I think is aware of penicillin. Penicillin is good for the soul, as we say. And the reason for that is because it protects us from different types of diseases. If we would have never been able to discover this from a fungi, we would have never had penicillin. Here, the yarn is for the dyeing, the color of the dye. Okay, just wanted to make that very clear. And then also, mold structure is very fascinating. So molds are multinucleated filamentous fungi composed of hyphae, and the hypha and so these hypha, which is the individual singular form, is a branching tubular structure. And what's interesting about this, it actually links units to the cross walls called septa. And the total mass of the hyphae is called the mycelium. The portion of the mycelium that anchors the mold and absorbs nutrients is called the vegetative mycelium, or composed of vegetative hyphae. So when you said hyphae, you're very much correct because these are very important for actually helping the mold to grow, basically. So that was a really good observation. And over to the right, we can see here aspergillus, which I really like a lot. I'm very familiar with aspergillus. Mold, this particular type of mold actually infects patients that have reduced immune systems. And so people that have HIV have been shown to be infected by aspergillus. And then there's two other types as well. And then we also are going to take a pause for the experiment. I want everyone to make sure that they're ready with all of their materials. We're not doing the bacteria experiment, but the bread mold experiment, okay? So these other two types of bread mold, the rhizophus, and then also the penicillium, these are also very used and very important because they can be found on breads and veggies. But penicillium can also be found on dairy as well as fruits. So it just depends on the type of environment that's around that the mycelium is able to attach to with a specific type of fungi to actually feed and for it to make it grow. So next, I'm going to hand this over to Dr. Marshall because she's going to lead the experiment today.
So what you should have are a couple of pieces of bread, your Ziploc bags, some paper towels, and some water because the mold needs moisture to be able to effectively grow. You'll need your tape, and you'll need your gloves and a marker to be able to label your different conditions. So does everybody have that at the table? So for the control condition with your gloved hands, I want you to pick up a slice of bread, hold it between your hands, then put it in your baggie, add a little bit of damp paper towel, seal the bag, and label it control. You can also date it today so you know when you started the experiment. All right, so for our next condition, you can take your piece of bread and choose to either wipe your keyboard or your computer screen. So you'll do the same thing when you're finished. You'll put it in your Ziploc bag, add a little bit of wet paper towel, seal it up, put your initials, date it, and then also tell what condition. So whether you did your keyboard or your screen. So for the third condition, I want you to find some place in the classroom that you think is particularly dirty and swipe your bread there. So that could be the floor, the wall, and the label is critical. So when it comes time to compare which condition had the most mold or the least mold, you need to be able to know which bread corresponds to which condition. Great, so for our next condition, I'll need you to take off your gloves and then hold the bread between your bare hands. You kind of rub a little bit, make sure you get nice and cover the surface of the bread. Then put it in your bags with your paper towel and label it hands. So for the last condition, I want you to either wash your hands with soap, if you have 20 seconds, or use hand sanitizer. Then touch the bread, and then you'll label this clean hands. And you can say whether you washed your hands with soap or if you use hand sanitizer. All right, if you got everything bagged up, everything labeled, you can set those to the side. And Dr. Hinton will come back on with our bacteria presentation before we do our plates.
So I really like bacteria. And so I added a slide about Serratia marcescens because it's my favorite bacteria that I actually first encountered when I was in undergrad at Winston-Salem State. And I was in microbiology with Dr. Pamela Jones, and she taught us about different types of bacteria. And I love this particular bacteria because it was pink. And so, like many other bacterias that are similar to Serratia marcescens, for example, they're called prokaryotes. They're single cell organisms that contain different type of um, particular organelles than what eukaryotic organelles would contain. And what's fascinating is a lot of people believe that bacteria or bacterium, if we're talking about a single bacteria, is actually taking itself and was engulfed by another bacteria that is now called a eukaryotic bacteria and made the first prehistoric, if you will, mitochondria. So another of my interests. So I really like bacteria for multiple things. I've actually kind of told you about the question already. I'm a little sorry because I'm actually really excited about bacteria. Did you know that bacteria is plural and that it should be pronounced as bacterium if we're talking about a single one? And they have been around for 3 billion years, so much longer than we have. And what's fascinating is they live inside of us as well. And so get this, on Earth right now, there are probably about 5 million trillion or 5 times 10 to the 30th power of bacteria. That's way more than what we ever could think we are, even if we got up to a 9 billion population on the earth. So there's nothing that compares to the power of bacteria. And we have got to learn how to have a mutualistic relationship with bacteria, not only in our gut, but in our environment. And when we don't have a mutualistic relationship, things happen. And that's the story that I'm going to tell you about before we talk about the good and bad bacteria. Interestingly enough, sometimes humans can be curious about different things, such as Serratia marcescens. What's really interesting about this bacteria is everyone knows that it's red, pink, and it has different colors of pigment based upon what's being produced in its genome. And what's fascinating is that at this particular picture right here, this is a scanning electron microscopy image, and actually shows that Serratia marcescens is a rod type shape of bacteria. And in the 1950s, which was uncovered by this blogger who works for Scientific American, Rebecca Kristen, she found that in the 1950s, the military actually was releasing this particular bacteria into the Northern California coast and area, and actually was looking for what would happen. So they tested it on the crew, and then they also tested it on other individuals in San Francisco. So that's actually near where Dr. Morton stays. So who knows, there might still be some Serratia marcescens there because of what's actually happened long time ago, because bacteria multiply very fast. And so what's interesting is that in September 26, 1950, the crew of the US Navy minesweeper ship spent 6 days spraying Serratia marcescens everywhere, everywhere. And they called this Operation Sea Spray. What's also interesting is that when they did this, the US military thought it was harmless. So this is why we need science first to test things, not based upon just some thought. We need to have factual driven experiments that are going to test a question and see if it's true or if it's not. And so when the military sprayed this not harmful bacteria, Serratia marcescens, the bug was mostly known for just red spots on producing it in food or infested food. So they thought nothing could happen related to clinical conditions. Well, afterward, after just 1 week, 11 residents were sent to the hospital at Stanford with urinary tract infections. And they're lucky because Serratia marcescens is known to cause other types of insults to the body. And if you notice, when we're staining on our bacterial plate that Dr. Marshall will talk about, this colors of Serratia marcescens may vary. So who knows, you may get something that grows that might be pink, and that might be Serratia marcescens. Or if we were using McConkey agar, it might be a different color. It'd still be a little pink, but not as much. So it really matters what type of plate we're using. And so everyone's using the same plate. So luckily we can do an experiment to determine if everyone's getting the same amount of bacteria that are gram negative or gram positive. And one thing I want to tell you about Serratia marcescens and why we need scientific principles and foundation for asking questions. And this is why you're really on the call, to discover your interest in science. Whether you stay in science or you do arts, you're still thinking about science because you're using a scientific method to create beautiful things that will share with the community. So in this particular space, Serratia, the S is kind of more like a space organism. And it sounds pretty cool, but it's really deadly. Serratia marcescens actually on its chromosome has this gene called AMP-C. And if it's expressed, it actually can neutralize antibiotic destroying beta-lactamase enzymes that will protect us. So meaning that if we eat bacteria, and Serratia has this ampicillin gene inside of it, it could actually cause for us to really be very sick because it actually develops multi-drug resistance. So it's very important to study bacteria and have a hypothesis in mind when testing it. And luckily for today, we do have a hypothesis. So Serratia marcescens accounts for only 1 to 2% of nosocomial infections. So I'm going to teach you this new word. Nosocomial infections is a sickness or some type of illness that you acquire from the hospital. But you have to be also aware that Serratia marcescens is deadly. Meningitis is nothing to play with. That's why we get a shot, a vaccine from that. So just be aware that bacteria can be harmful and it can be deadly if we're not careful about answering critical questions. So enough about the bad bacteria. Let's talk about some good bacteria. Well, bacteria that are good are everywhere on our bodies, inside and out. And as you can see from this very cute cartoon, sometimes you have to think of bacteria as being friendly. Not all of them are bad. Interestingly enough, inside of our gut, there's 500 to 1,000 species that live there. And they help us to break down food. And they also help us to protect against our blood pressure, help us protect against getting fat in the context of obesity. When there's extra adiposity, it actually helps us to protect against diabetes. And there's still stuff that needs to be discovered. Scientists call the bacteria that live inside of us in our guts, our intestines, called the microbiome. And what's also fascinating about these bacteria, they mutualistically work to help protect us. So on the outside on your skin, the rest of your body is covered with microbes to help boost your immune cells. And so this really is a protective thing. So we have to really think of bacteria not being only bad, but also good. And interestingly enough, we haven't talked enough about the bad bacteria just quite yet. So bacteria can cause disease. And we also have to be careful. So if we're using hand sanitizer always to do something, you know, we're maybe destroying the good flora on us, or it could be the bad flora that are on us. So we have to be aware of what we're doing. Also, some particular types of bacteria that are actually common bacteria that you hear about in the news, E. coli, Salmonella, they are healthy for us and they live inside of us, but they also can cause opportunistic infections or pathogens because they can contaminate different types of food or drinks. So sometimes when we're actually at the store and we get our veggies from the US or abroad, there's infections that happen because there's some contamination because people don't wash the actual bacteria off, or it has gone inside the soil. So now the actual bacteria have been uptaken inside of the veggies that we're eating, and that's why we're getting these sicknesses. So one particular common thing that you can think about inside of our nose, our mouth, our skin is streptococcus. And it causes many different types of illnesses, such as strep throat, fever, and then other types of toxic shock syndrome, which is a little scary. And then also bacteria can be really ugly. Bacteria, if we don't use our antibiotics properly, we actually can cause for different things to occur with our system, which means that the bacteria could basically take over and it's resistant. So that was kind of an introduction with Serratia marcescens. Also, there is a growing list of infections that bacteria can cause. And one we're all very familiar with, pneumonia, or walking pneumonia even, or tuberculosis. Some other things could be blood poisoning, which is very dangerous, and gonorrhea, which is more of an STD or STI, and then also foodborne diseases. So we have to be very careful about how we think about using antibiotics. If we have antibiotics that are prescribed from the doctor, not for a viral infection, but for a bacteria infection, we should take the course for 2 weeks, not just 1 week and cause it done because, oh, I'm feeling better. HA is now can go ahead and play with his friends. Or in this case, since we're living in COVID world, we are on Zoom and hang out and do some writing. Well, that's not good enough. You need to take the 2 weeks because if you don't, you could actually build the bacteria up to come back again. And then now it has antibiotic resistance from what was treating you to make you feel healthy again. Also, one particular bacteria that's like this is MRSA. So basically it's a Staphylococcus aureus that is methicillin resistant, meaning that it's drug resistant to this particular type of drug that actually would mostly destroy most Staphylococcus aureus. And so this is important, like I remind you. So go home today, talk about this with your parents and your siblings. Tell them that the importance of finishing antibiotic resistance is the one key thing that you learn from all of this PowerPoint. And tell them about the cute bacteria, Serratia marcescens. And so there are different types of bacteria. There are actually 5 different classes. There's spherical, rod, spiral, comma, and corkscrew. And as you can see, there's an example of each one of these. And so one that I'll just focus on, I like bacilli, which is rod shaped. And so you can imagine here, Bacillus cereus, that is what the rod shape looks like. And so when you're able to review the PowerPoint again, you'll be able to see you'll be able to see all the different types of shapes and you can match them up. And then also bacteria are classified by a cell wall. That is a gram-negative or a gram-positive bacteria based upon their cell wall. Gram-positive bacteria have a very thick cell wall. Gram-negative actually has two membranes in between their cell wall. And so what's interesting about this is that the cell wall was very, very distinct, and it's made up of these particular sugars called peptidoglycan. And what's also interesting is that there's more peptidoglycan in the positive than in the gram-negative. So in 2 weeks, we'll actually be able to see what happens with our bacteria that grow in the Petri dish, and we'll be able to stain them. So that's going to be even cooler because we'll be able to distinguish between gram-negative and gram-positive. And this technique that we're talking about in the context of staining bacteria was developed by a Danish bacteriologist named Hans Christian Gram. So that's where this technique came from, called gram staining. So Dr. Marshall will take over. She's going to talk about streaking our plates, and I'm going to zoom out so that she can focus on, so you can focus on her.
So what we're going to do now is get our agar plates, and we are prepared to streak some bacteria. So what you'll need are your agar plates. You'll need a Sharpie so that you can divide the plate into 4 different quadrants, because we're going to test 4 different conditions. You will also need gloves, cotton swabs, and a bacterial loop if you've got one. Okay, so the first thing I want you to do, I don't have a plate with me, but I have a drawing, is divide your plate on the back like this. So you'll draw a line straight across and then straight up and down. And then at that intersection, I want you to label 1 through 4. And you guys can ignore that swiggle for now. The other thing you can do is somewhere along the plate, write today's date and your initials so we know which one is yours. And we're doing all of this on the back side of the plate. So the first thing I want you to do is take a clean swab, make sure it's clean, and I want you to swab the inside of your cheek. Once you've done that, I want you to go in your first quadrant, gently touch the tip of the Q-tip to the agar, and make a swirling, not a swirling motion, but a zigzag motion from one corner to the other corner of that quadrant. So kind of follow like that drawing. And that is the same type of sweeping motion that we'll use for each condition. The agar is firm, but it's still soft. So you want to be gentle as you streak the swab across it. You don't have to press hard to get the bacteria off. And somewhere to the side, make a note that's what your conditions are. So you'll put condition 1, cheek. So for your second condition, I want you to take out your cell phones, and or you can swab the computer screen if you don't have your phone on you. So you'll do the same thing, take the clean Q-tip or a clean swab, kind of streak and rub real well on your phone or on the computer screen. Then you will gently pick the quadrant number 2 on your plate and streak the swab across the plate. So for these, I want you guys to get creative. Different locations, either inside or outside the classroom, to swab and then streak your plate. I will ask not to rub it on yourself or on somebody else. So pick some inanimate object to swab. So to me, this is one of the most fun parts of the experiment, just to see how creative you guys get. So you'll take the Petri dish face down. So it has the top and the bottom on the top. You'll take a thin strip of the parafilm, remove the little wax paper part, hold one end, and then along the edges, you'll stretch it. So it will form a seal right around the edge where the top and the bottom of the plate. And then if you can, when you're finally finished and the plates are wrapped, you can stack them in just a warm, out of the way place in the lab and let the bacteria grow. They're pretty good at just doing their own thing. We'll say that this part of my phone is the top, this part is the bottom. So I'm going to hold the plate like this, keeping the top is on the bottom's on the top. what you'll do with a piece of your parafilm, remove the paper, the wax paper backing, and just hold the film. You want to hold it to one edge of your plate. And then what you're going to do is wrap the parafilm around and stretch it until it completely encircles your plate along that edge. And you'll see that as you pull the parafilm, it will stretch and it will cling to the sides of your plate. And that's what you want to do all the way around. And once you make one complete circle with a little bit of overlap, that's all you need. You don't have to wrap it 3 and 4 times. All right, once your plate is wrapped, it just needs to be set in a warm place to the side. And that is done until we come back and stain our bacteria. Thank you for having us.